This is my aging slide, just reminding me that actually antiretroviral treatments is not a cure, but it did really suppress virus replication and increase the spans of life in the patients, and of course the comorbidity associated with the presence of the virus and or in combination with the treatment which goes on with it. So the problem that we need to resolve in order to cure the disease is either identify the infected cell in the reservoir, kill the cells with the virus in it, or save the cells and get rid of the virus. So the strategy that we used was the latter. So this basically cartoon illustrates that that the chromosomes after the infection carries a viral genome, integrated copy, either functional or pieces of viral DNA. So antiretroviral DNA does not, anti-ART does not really eliminate the virus from the chromosome, nor does suppress expression of the viral genome. What it does is inhibit viral replication. So some of those problems that you see, either cardio or brain or some other area, might be associated with the toxicity associated with the virus in the cells, which continuously probably express, although at the very low level, some of the protein, viral protein, which probably do have a toxic activity. So we employ the CRISPR-Cas9 gene editing strategy. It's a very simple technique. And actually, we started on that about 4 years ago. And the component of the CRISPR-Cas9 is an enzyme by the name of Cas9, which can introduce a nick on the double-stranded DNA. And this Cas9 can be basically programmed to identify the specific region in the DNA and introduce the nick. And that the RNA is so-called the guide RNA. So what you could do is you could actually develop the guide RNA, which is complementary to the gene of your interest. For us, HIV-1. So once you have a Cas9 and the guide RNA, this complex identify the gene of your interest and introduce nick. So what happens is after that is that nick, which is introduced by CRISPR-Cas9, can be repaired by non-homologous enjoining DNA repair pathway, which every cells do have that one. And then, but the thing here is that this non-homologous enjoining is not the best repair pathway. The fidelity is not that much. So what happens is that when you repair, the nick introduced mutation, either base substitution or insertion deletion mutation or indel mutation. So the outcome is you create a mutant virus. The mutant virus can escape from the initial treatments. So you basically can create mutation, which still replicates. And if you have indel mutation, the escape frequency is low. What we did in the laboratory, instead of using one guide RNA, we target multiple regions within the HIV genome. The idea was that if we do that once, so we can probably cleave here and excise viral DNA and rejoin the rest of the chromosome, again through the non-homologous enjoining, large deletion, no probability for escape. So that's what we call the excision strategy. So with that in mind, now I'm going to show you 4 sets of data. One is one is done in the cell culture models in the laboratory, latency model, and the other one is the patient samples, and then 2 sets of the data in the 2 small animal models. So we have in vitro, ex vivo, and in vivo stuff. So for the in vitro experiments, we use the latency model, cell culture model developed by Jonathan Carn. And what John has done, he took the cells, CD4 T cells, and introduced viral genome which has a NEF deleted, and instead of that, we put the GFP protein as a marker protein, okay? So this is a LTR for HIV. So this is basically shows the U3 region of the LTR. And these are the area of the LTR that we target. So remember that HIV has LTR in the 3' end. And this sequence at the 5' end is also replicated in the 3' end, all right? So, so now we have a cell line, CD4 plus T cells, which contains this viral genome integrated in the chromosome, but it's a silent stage, very similar to what you see in the patient sample, in the reservoir. The virus does not replicate, does not express. So this shows here. So if you take the cells, which is contains this one, in the normal condition, virus is not expressing, but you can stimulate expression of the virus with a PMA or tercostatin A or SAHA. This is a HDAC inhibitor, okay? And you see the majority of the cell, greater than 94% of the cells right now is green, indicative of the virus gene is turned on. Now, if you introduce CRISPR-Cas9 system in the cells under the condition that untreated no SAHA, no PMA, the same. And if you introduce PMA and TSA, nothing comes up. So that was the first indication for us, illustrating maybe that the CRISPR system is working in the in vitro system. Then we went on, tried to do some genetic analysis. So I'm going to skip a lot of the data because of lack of time, but I'm going to show you this. This is basically ultra-deep whole genome sequencing of the cells treated with the CRISPR-Cas9 system. So basically what we did in this one is we sequenced entire whole genome of the cells, and this is untreated cells, and every single nucleotide on the cells were read 100 times. So you could imagine 6 billion cells, nucleotides read 100 times, and those information were gathered bioinformatically and presented here. So what you see here, these are the reads, basically. You see that you're reading a HIV genome here, okay? And this cells has a Cas9, but not guide RNA. So you're not editing viral genome because it doesn't have a guide RNA. So, and this is the one who received a Cas9 plus guide RNA, which recognized HIV, gone. There is no single read here which representing the HIV is present. What you see here is a residual of the LTR. And then that's basically the guide RNA and the primer which was used basically outside of the LTR. And this is really non-pathogenic, small segment of the LTR. So having this data, then we move on, we did a chromosome walking. And we want to see where the virus DNA is incorporated and how many copies of the viral DNA is present in the cells. So the first one, we identified that one copy sits in the chromosome 1. And the specific region in the chromosome 1, and that's the structure of the virus DNA incorporated here. Now, having the data about the deep sequencing, then we identified the region outside of the virus, and then we try to amplify the gene, viral DNA, in the untreated cells, and this is a DNA which we amplified in the gene, in the control. And we try to amplify it in the treated cells, nothing. And we got a smaller DNA fragment. You take this one and you sequence, you realize that the CRISPR system nicely eliminated cut in the B domain, guide RNA B, in the 5' LTR, and then B domain in the 3' LTR, remove entire viral genome, and basically converted the cell, which had a virus in it, became a virus-free. Then we look and we found out, aha, there's another copy of the virus, and that is in the chromosome 16. There's 2 copies in the cells. But in chromosome 16, the position is different, and we did the same, and we did a chromosome walking. We realized that in the chromosome 16, guide RNA A picked up from 5' end and B from 3' end, 3' LTR. Remove that into control. Again, you see full-length basic viral DNA, not in the treated cells. A smaller fragment shows the sequence. So that was the first time, show that you can actually eliminate the viral DNA from the chromosome, and you convert the infected cells, become completely uninfected cells. There's no evidence, there's no way that this, basically there's an escape on this one virus and they replicate. And then excised DNA, viral DNA, disintegrates and disappears with less than 48 hours. We searched, and we never saw any footprint of the excised DNA in the viral DNA in the cells. So we did a lot of bioinformatics and then off-target assay. I'm not going to talk about that. It's going to take probably another 45 minutes. We did all the bioinformatics, and we rule out any side effect associated with the excision of the viral DNA on the host genome. No toxicity, no impact, no inducing the apoptosis, no cell proliferations are affected, and no autophagy, none of those things, no cell cycle changes. So safety was actually verified in the cell culture model. Then we asked the questions whether or not the presence of the CRISPR in the cells can protect the cells against a new infection. The answer to the question was here. So what we have here is basically we selected 4 different clonal cell lines. One clonal cell just expressed Cas9. The other one expressed just guide RNA. See, Cas9 is missing, guide RNA is here missing, and guide RNA is here. Cas9 is here, guide RNA is missing here. Then we selected a 2 cell line with different extent of the Cas9, but equal amount of guide RNA. We infected with HIV. And we realized that the cell, which has a Cas9, not guide RNA, or guide RNA, no Cas9, can be infected. But the cells, which has both of them, become protective against the viral infection. And actually, if you see, the protection is a little bit better here, which has more Cas9 than the AB9 clone, which has a little bit less than this one. So suggesting that this technology should get into the clinic, not only can be utilized for the treatment, has a prophylactic legs, can actually protect the cells against the viral new infection with the HIV. So then we went on to the patient samples. I'm going to show you a very limited study, but this is published already. So we got the patient samples, and from the 4 cases, this is the clinical information about the patients. We propagate the cells, and this is a procedure, expanded, and then they introduce a CRISPR-Cas9 system using a lentivirus in this case. Okay? And this is the results. You see that the induction of that in the PBMC or CD4 cells significantly suppress virus copy number in the cells. So this is basically a couple cases, not in all of them. In some of one of the cases, we see that the suppression is not as good as here, but there's something that needs to work out, whether or not in this particular case there's variation of the virus, which our guide RNA doesn't recognize, or some efficiency of introduction is, we still don't know that one, but obviously there is a way to get around this one. We're trying to see whether or not we need to personalize our CRISPR-Cas9 system, which completely eradicate the virus in this patient as the same way as it almost did it here. Then we move on, we want to do the in vivo experiments. For in vivo experiment, we need a delivery system. So we first focused on the AAV9, adeno-associated virus. There was a couple of them, and a couple of serotype of AAV. One of them was AAV6, 9, DJ, and DJ 4 times. And what you see here, the band is excision. suggesting that this AAV can deliver the virus, the CRISPR-Cas9 into the cells and excise the HIV genome. So then we went on and we used 4 different, basically in vivo animal model, as I explained. The first 2 were the transgenic mice, TG26 transgenic mice and transgenic rats, and then we use eco-HIV and BLT humanized mice. Let me just show you one of the transgenic mice. Here is basically shows the transgene in the animal model. And then here we have in the transgene, there is a piece of the gag and pol is removed, and that's what the virus does not replicate, yet the LTRs are there, and the viral genes are here and produce TAD and envelope. And this is a guide RNA that we use. In this case, we use guide RNA recognizing LTR and piece of the gag genes. First, we did ex vivo. We used a cell line from the mice, but introduced a CRISPR-Cas9 system by the AAV, and detection of the band at 345 is indicative of excision. The subsequent experiment, after this, we went on, we did the IV injection, but in this case, we used different primers, and with notion that if there is excision, the fragment of the 160 nucleotides will appear in the gel analysis. So, and this is the results. There should be something. Okay. So, this transgenic mice, IV injection, tail vein, tissues were harvested, genotyping was basically performed using basically a PCR, and then the critical lane is the 2. First and third lane is the controls. The appearance of 160 nucleotide in size fragments you see across everything is indicative of the excision. And we verify actually this one by sequencing. We verify that one by sequencing. The top here is a control that you see the viral DNA, the viral DNA is present, and the other ones are the excision. And the excision is so precise, exactly the same way that you target from the guide RNA A to the gag genes. And this is basically the region which is removed. And this region, by the way, is not small, it's about 1.3 kb. We completely eliminate the capacity of the virus to replicate or express the gene. We did the same thing in the rat model. And here it shows the size of the excision. Top band is a control, and here are basically shows that the fragments are missing. And we look at the gag RNA, the remaining of the gag RNA and the envelope in this case, and you see, look at that, the suppression of the virus RNA upon the treatment. So suggesting for first time, showing that you can actually take the CRISPR system and introduce in the cell by IV, and it works. It gets every cell and the tissue, and they eliminate the viral DNA. Then we moved on. Scott, how are we doing with time?
We're a little bit over, but keep going. Maybe, how many more slides do you have?
I have about 40. No. You ask. So let me just go through a couple of these. Then we did, this is basically eco-HIV. Eco-HIV is a HIV which has a GAG, GP120 was exchanged between 120 for HIV to murine leukemia virus. David Wolski actually created that, and this virus replicates actually in mice. So we took that and put the basic luciferase gene in the virus. So every time that you inject the virus replicate, you can do imaging, and you see that that's the site where the virus is basically present. And this is a control. You see here, dorsal and ventral imaging. And you see the virus present is all over the place. Probably this is a lymph node, at least the imaging guy told me. And then this is the time that we introduce in Cas9 guide RNA. You see this begin to clean up after several days. And this is basically showing that in lymph nodes, in the treated cells, in untreated cells. But that's not enough. We went on, we took the DNA, and then we sequenced actually, and we show that again, we show that the basically size of the eradication in those tissues from the rat model. And this is another early sequencing. This is, again, an area that which shows that the elimination of the viral DNA in the cells. Then the next one was, for us, we use VLT mice. And this is a humanized mice. And then we infected with the cells. And then with the virus, the first step for us was just look at the spread of the virus in the animal model. And as you see here is that the virus is spread. And these are the tissue. The presence of virus is detected in the kidney, spleen, liver, and a few other places. And then we went on and we introduced, but the interesting thing here was that after a while, expression, you see the extinction of the virus expression. But the virus is there. Viral DNA is there. So when you introduce AAV-Cas9, in this case, these are the animals. And we did harvest the DNA and look, and that's the excision. Suggesting that in the BLT, you can actually also excise viral DNA. Now, as you know, that the real task comes as a rebounding, whether or not that when you stop antiretroviral, the virus comes back or not. I don't have that data for you, but I do have a system in place. So if Scott invited me about 2 months from now, I probably will show that it's working or not. The experiment is a process, but this shows that the procedure that we are using, basically infection, and then the antiretroviral, we draw, and then before we draw, we introduce AAV, Cas9, and we let it go. In the control samples, which there is no AAV, you see the rebound is happening in the mice. Okay, but now experiment is in process that we are basically injecting AAV here and see whether or not it's coming out or not. So that's all I have right now, and these are my collaborators that help me out to, actually it's a truly teamwork, and these are the group at the Temple, and these are the different groups and different parts. You know, from Drexel, Brian is doing a lot of genotyping for us. George Mason, Fatah Kashanji is working on some of those humanized mice. And we have Dr. Madhavan Nair. We are introducing a nanoparticle, using nanoparticle as a tool for delivery. Nebraska also, Gendelman is helping us with the different kind of nanoparticles. And Jonathan Korn made those cell lines. We're collaborating on the latency. And in South Carolina, collaborating with Rosemary Booth. Rosemary Booth is interested in neurocognitive disorder, behavioral disorder. And actually those rat model is something that she's working. We send our material, so Rose introduced into the rat, and then the result that showed was done actually in the different laboratory with our reagent. But it was comforting, showing that reproducibility in different hands. But Rose is interested in looking at the neurocognitive. And she's telling me, actually we're putting a paper together, that before and after eradication, after eradication, there's significant improvement in neurobehavioral in the transgenic rats. So that's tell us about something. Maybe we should just begin to think about restoration of the post-eradication. And so that's something that we are working with Rose. So I'm going to stop at this stage, and that's about it. Thank you.